So it's 4. I think we can start. So welcome everyone. Thank you for being here at this for this month ISV Next Generation Vaccinologists webinar series. So today we're gonna have 2 very nice talks coming from 2 young researchers. Actually, both are PhD students. And they're gonna show us some very cutting-edge medicine. So we will first have Nicholas Tursi from the University of Pennsylvania Perelman School of Medicine, and then Ilke Aernout from Ghent University. So first of all, let me just make you a little bit of publicity regarding the ISV Next Gen group that you should 100% join. 1 second. So why should you join the ISV group, and in particular the Next Gen group? So first of all, you will have a reduced registration fee for the ISV Annual Congress that this year will be in Stellenbosch in South Africa. And also you will be able to access to the grants that will cover everything for the Congress, so consider. Then you will be able to access the career mentorship program, then participate in various ISV committee, but especially know a lot of people, and so get your a big growth in your network. So I really suggest you to subscribe. And in particular, the best thing is that for postdoc and students, 1-year membership is only $35. If you want to follow more closely what we do before subscribing, please don't hesitate to scan the QR code for our WhatsApp and our LinkedIn profile, or don't hesitate to write us on our email. So said that, I would like to introduce you our first speaker. So our first speaker is Nicholas Tursi, as said before. He's a PhD student in immunology. The University of Pennsylvania Perelman School of Medicine. He's in the lab of David Weiner at the Wister Institute Vaccine and Immunotherapy Center. And his research is based on new lipid-based DNA formulations for infectious disease prophylaxis. So Nicholas will talk first, and if you have any question, just write it down on the Q&A below, so that at the end of the session, I can just read them out so we can answer to all your questions. So don't hesitate to ask. The most question, the better. Please, Nicholas, the floor is yours.
Thank you so much, Allegra. Hopefully you guys can hear me okay. My name is Nick Tursi, as Allegra said, and I'm excited to share some ongoing work we have in the lab on lipid-based DNA formulations for infectious disease vaccines. And so the Weiner lab primarily focuses on DNA vaccine technology or DNA-encoded biologics. And DNA within the genetic vaccine space has a number of different opportunities and some challenges. Firstly, the DNA is very fast and flexible in terms of encoding any sort of antigen of interest from a variety of pathogens, and really going from just simply sequence into plasmid into delivery can be quite fast. DNA itself is cold chain independent, non-live, non-replicating. It can induce both cellular and humoral immunity, and that's kind of one of the important facets of the platform, is its ability to induce CTL responses, and that's been demonstrated preclinically a number of times with a variety of different antigens, as well as in clinical settings. And this is just an example of a neoantigen vaccine in hepatocellular carcinoma, where DNA vaccine here delivered by electroporation delivered really strong results here in this phase 1 clinical trial. And also, as I sort of alluded to in the context of biologics, DNA can deliver these biologics with a pretty long-term expression profile relative to, say, recombinant protein delivery. In the context of some challenges, currently DNA vaccines primarily require physical transfection modalities such as either electroporation or jet delivery or gene gun to be able to really induce strong levels of transfection in vivo. And additionally, DNA has not necessarily elicited the same sort of serological potency relative to other platforms. And so this is an example of a clinical doses of either a DNA vaccine encoding SARS-CoV-2 spike or the BioNTech mRNA vaccine. And so although the T cell responses are stronger in the DNA context, there still is a gap in neutralization response here shown on the right. So one of the things that we've been interested in is trying to improve that serological profile of DNA vaccines while still maintaining the sort of benefits we have in the context of T cells. And thinking in terms of alternative delivery modalities, lipid nanoparticles are, of course, crucial in the mRNA space, both in the context of delivery, but as well as in the context to actually adjuvant the immune response. And so the top figures here is just demonstrating that the ionizable lipid is pro-inflammatory, even without the RNA payload. So this can be inducing some inflammation that would be important for priming an immune response. Also, empty LNPs themselves can actually adjuvant recombinant protein vaccines. And so this is an example comparing recombinant protein with a squalene-based adjuvant here in blue to either empty LNPs in green compared to an mRNA-LNP in red. So the empty LNPs themselves have a pretty robust adjuvant capability themselves. And so we were interested in trying to utilize lipid nanoparticle delivery for DNA vaccines, and that could have an advantage considering that mRNA synthesis starts with plasmid DNA, so you'd be able to skip a lot of the kind of costly synthesis steps for mRNA if you were to utilize plasmid DNA. And so this critically could be of value if it retains the positive product profile of DNA vaccines such that it's temperature stable and et cetera, induces long-lived immunity. It's dose sparing relative to traditional DNA delivery because, say in the clinic, we are giving in the scale of close to 1 milligram of DNA. And the ability for it to be device-free would also provide additional options in combination with or separate from the current delivery modalities. Whoops. Okay, so this is kind of a summary of some of the at least initial steps we've taken in terms of trying to reduce to practice these ionizable DNA LNPs as a vaccine modality. And so the problem is that LNP formulations for RNA are not necessarily, excuse me, going to be suitable for plasmid DNA. And that may be because of the relationship between the ionizable lipid components and the nucleic acid itself. And so that's termed the N to P ratio. The N is the tertiary amine groups in the ionizable lipid that this is kind of pictorially in this cartoon represented with these green lipids here, interacting with the phosphate bone of the nucleic acid. And so RNA uses specific N to P ratios, and we hypothesized that we would need a higher N to P ratio to better accommodate the DNA plasmid, considering it's double-stranded and much larger, so it has a lot higher number of phosphate groups. And so we tried various N to P ratios, and here's an example below of a higher N to P ratio of 10.5 through a low N to P ratio of 2.6. And so just to illustrate, we have more favorable size characteristics with our high N to P ratio, which is a smaller size here, closer to say 70 to 100 nanometers, and a zeta potential closer to 0. And these biophysical characteristics kind of are indication of particle stability. And when we look under the microscope, we can see that these DNA LNPs, and this is at our high N to P ratio here, look like lipid nanoparticles by microscopy. And so importantly, that we wanted to next understand whether our high N to P ratio formulations would indeed actually improve the immune response, as well as conferring these biophysical advantages. And so here I'm using the 2009 pandemic H1N1, the California 2009 HA as a immunogen, and using the germinal center as an immunogenic readout 14 days post-immunization. And what I hope you can appreciate is that the higher N to P ratio formulations here induce a much stronger germinal center response relative to the low N to P ratio formulations here in green. So this is at least supporting that the high N to P ratio formulation is indeed superior to these low N to P ratio formulations for plasmid DNA. And so from here, we down selected on this blue formulation for the rest of the talk. So at least to start looking at innate immune activation after immunization with these DNA LNPs, So we're comparing our DNA-LNPs to mRNA-LNPs at the same dose or to recombinant protein and Adavax. Adavax is a squalene-like adjuvant, which is similar to MF59, which is in the approved fluad vaccine. And so looking initially at a variety of different innate immune populations, but just for the sake of time, focusing in on migratory dendritic cells. What I hope you can appreciate is there's a nice improvement in the migratory dendritic cell response after DNA-LNP immunization here in the draining lymph nodes, most notably among this subset on the right, the CD103 positive MDCs. These migratory dendritic cells are similar to CD8 alpha positive resident dendritic cells, so they're important for cross presentation and priming of CD8 T cell responses. And so we thought that this might be an early indicator that we might be inducing strong CD8 T cells with our DNA-LNPs. And across these populations, looking at activation using CD86 as a marker, our DNA-LNPs induce much more robust CD86 upregulation as compared to either mRNA or adjuvanted protein. Considering the innate immune activation profile we saw, we wanted to understand how these DNA-LNPs are being sensed. And so briefly, we thought that this could be primarily through two mechanisms, one being through either Toll9, or which would sense CpG motifs, or through cGAS sting pathway, which would be sensing double-stranded DNA. And so we developed an assay in-house using inflammation as a readout to try and demonstrate this. And so what I would like to show you is that if we use inhibitors to different parts of the cGAS sting pathway, either to cGAS itself, to sting, or to tank binding kinase, we completely ablate signal in this assay, as opposed to if we use an inhibitory CpG, we don't lose any signal compared to our DNA-LNP alone. So this suggests that these DNA-LNPs are being sensed through cGAS sting. And for the sake of time, I omitted this, but we also looked in vivo in using sting knockout mice, and we confirmed this phenotype using our innate immune activation, as I showed you in the previous slide. We had the opportunity to do some single-cell RNA sequencing to try and further characterize these innate immune responses. And so briefly looking at a variety of different innate immune populations in the draining lymph node, we can see that among genes that are upregulated in the DNA-LNP relative to the other groups, broadly, there's this pro-activation as well as a pro-migration signature in a number of these innate immune subsets, specifically among neutrophils and monocytes, strong upregulation of these chemokine genes or chemokine receptor genes. And so we think that overall, the DNA-LNP uniquely is inducing these specific chemokines to help try and prime immune response. Next, looking in the context of serum titers, importantly, this was one of the primary goals of this project, was trying to induce particularly robust serum IgG titers. And so the immunization scheme is the same as before. We're priming with either our DNA-LNP as compared to mRNA or adjuvanted protein, and monitoring the serum response over time. And so we observe really robust serum titers here in the top right to the matched California 2009 HA. That is comparable to both mRNA and adjuvanted protein. The response is equally as sort of broad in terms of its binding titers against mismatched HAs. This is to the much later Guangdong 2019 HA or Victoria 2022 HA, as well as in terms of antibody functionality in terms of overall HAI titer, and this is to a near match virus, this Netherlands 2009, or to a more recent clinical isolate, this New York City 2022. So across the board, we're eliciting pretty robust serum titers in terms of both overall magnitude, in terms of breadth and functionality. So next, we wanted to understand the CD8 T cell response among these adaptive populations. And so looking in the spleen, 14 days post immunization, we observe really robust CD8 T cell responses with our DNA-LNP. And so this is looking at interferon gamma secretion in effector CD8 T cells in these FACS plots here and quantified on the right. And so with our DNA-LNP, really robust CD8 T cells superior to that of either mRNA or adjuvanted protein. This is also true amongst different effector molecule or cytokines among CD8 T cells, either CD107, which is a marker of degranulation, or TNF-alpha. Amongst CD4 T cell populations here in the bottom right, we see pretty comparable responses between our DNA-LNP and mRNA-LNP in terms of interferon gamma secretion, TNF-alpha secretion, IL-2 secretion. So we're inducing, at least among the effector CD4 population, pretty comparable responses between our DNA and mRNA, but superior responses in the CD8 T cell compartment. So we're not losing anything amongst our CD4 T cells by priming these CD8 T cells. Importantly, for any kind of vaccination platform, inducing a strong memory response is crucial. And so we had the opportunity to prime mice and look just over a year post-immunization at different memory, either T or B cell populations. And so these two figures here are looking at memory T cell populations, either by interferon gamma secretion by ELISpot or by flow cytometry. And so even just after a single dose, we still are observing really robust effector memory CD8 T cells specific for Cal09 with our DNA-LNP. And we see pretty comparable antibody secreting cells in the bone marrow here. So we know that the responses that we're priming in the serum are indeed homing to the bone marrow, as well as memory B cell populations in the spleen. And so between our DNA and mRNA-LNPs, we see pretty comparable memory B cell responses specific to Cal09 HA. And that's even slightly better than what we see with recombinant protein in Adavax. So we had the opportunity to take this into a slightly larger model in New Zealand white rabbits. And so here we're increasing the dose to 50 micrograms and comparing either naked HA DNA to our DNA-LNP or mRNA-LNP. We use a prime boost regimen at day 0 and day 28 and looked at the immune response over time. And so initially 2 weeks post of boost, looking at T cell responses in peripheral blood, our DNA-LNP is inducing stronger T cell responses in this rabbit model that do slightly wane over time, but ultimately at a memory time point, we still see that past 200 days post immunization that we still have robust T cell responses with our DNA-LNP. Amongst serum IgG titers, we see robust serum IgG binding titers with both DNA and mRNA, and the DNA even out around 200 days is even slightly better than the mRNA, and that's quantified here on the right. So we know that we can at least scale up our DNA-LNPs into a slightly larger model in New Zealand white rabbits. And finally, so we had the opportunity to do a lethal challenge, and here we're actually changing the antigen to SARS-CoV-2 spike. Comparing either a DNA-LNP to an mRNA-LNP at the same dose. We primed the mice once and we looked at serum titers 8 weeks post-immunization, and that's binding titers are at the top here against the matched RBD or full-length spike, or to mismatched delta spike or the BA2 spike. Overall, robust binding titers pretty comparable between DNA and mRNA. We also have competitive neutralization titers as well as complete protection in this lethal challenge model from both morbidity and mortality and this lack of clinical scores. So what I hope you can appreciate is that our DNA-LNP formulations do indeed exhibit pretty robust immunogenicity in vivo after a single dose, competitive in terms of serum, antibody titer, functionality, and breadth with mRNA and adjuvanted protein, as well as inducing strong memory responses. They're protective in challenge. And so we look at these DNA-LNPs as a way of kind of complementing other DNA approaches in this sort of DNA toolbox, either using various physical delivery modalities as well as these LNPs as kind of another angle of advancing DNA vaccine technology. And so this was a large collaborative effort, and so take the time to thank all of my collaborators here, and I'm happy to take some questions.
Thank you very much, Nicholas. This is very, very impressive. So just a reminder, if you have any question, just write it down in the Q&A session so I can ask them to Nicholas. So I already have one. So it's Margaret Liu that asked, thanks for the great talk. When Feltner and Al first published on DNA plasmids, they specifically could not get better delivery in vivo than unformulated plasmid with their formulation. What is different about your LNP formulation that you were able to achieve this breakthrough?
So I think primarily it's due to the higher NTP ratio formulations here. So I think we're better accommodating the DNA plasmids, improving overall stability and biophysical parameters that's helping to get delivery. So the LNP is also serving as has its own intrinsic adjuvant activity. And so in this way, also the way we're delivering the DNA, it's inducing CGAS, sting-dependent inflammation. So I think there's this kind of synergy between the DNA and the LNP here in this context.
Okay, thank you. I have another question. So you already have a lot of questions. So we're not going to go over all of them at the moment. We will. I will just take 2 more questions, and then we will continue at the end of the talk of Ilke. So Makram Safi asks, did you assess the risk of DNA integration into the genome?
Not with this platform, but we and others prior had done many studies in the looking at DNA integration, and we did not observe any significant levels of DNA integration, at least through either EP delivery, and I would assume these studies have been done with other delivery modalities as well. So not specifically in this context, but we wouldn't necessarily expect this here either.
So last question, is there a way to reduce potential waning of immune response over time?
Is there a way to, I'm sorry?
To reduce potential waning of immune response over time, I think reduction. Waning? No, I'm not sure what he means. Well, I will go maybe, yeah, okay.
No, please, sorry.
You understood the question? I'm not.
I think so. I mean,
Yeah,
I will
okay.
answer the way I understand it. So I think part of the waning kind of observation that people have observed, at least with some of the clinical mRNA formulations, I think that probably has things to do with priming. I mean, this is just a hypothesis of mine. So I think if we're able to prime the kind of right populations, and perhaps this is what we're seeing here with our DNA LNP, at least the immunogenicity in rabbits was very encouraging, but of course, to really understand this in larger models, we'd have to scale this up beyond rodent models into things like non-human primates.
Thank you very much, Nicholas. So stay tuned, everyone. We will continue with your questions at the end of Ilke's talk. So I will switch then to Ilke. So Ilke Aernout is a PhD student in the vaccine delivery group in the Ghent research group on nanomedicine at the University of Ghent. So she's also my colleague. And so she works on adjuvanted mRNA-LNPs for vaccination against multi-drug resistant intracellular bacteria. So in particular, she works within a project called Vaccinologists, where they develop novel mRNA vaccine against Mycobacterium tuberculosis. So Ilke, please, the floor is yours.
Thank you, Allegra. So thank you for inviting me to this webinar here today. In today's presentation, I will explain you a bit more about my work on Galsomes. It's an adjuvanted mRNA-LNP platform, and we aim here to develop it for as a vaccine against Mycobacterium tuberculosis. So the Galsomes, it's adjuvanted mRNA-LNP platform, and it looks quite similar to the conventional mRNA-LNPs. The only difference is that it includes an extra adjuvant. It's called the alpha-galactosylceramide, or alpha-galcer. Our formulation usually consists of 2 major components. It's a C12-200 LNP that we often use. And we first co-encapsulate nucleoside-modified mRNA, encoding the antigen of interest. And we use here nucleoside-modified mRNA to produce an immunosilent response and to limit the type 1 interferon activity, and in this way increase the stability and translation capacity. On the second hand, next to the mRNA, we include our adjuvant, which is present in the lipid structure actually, and it's a glycolipid antigen that is presented in a CD1d pathway, and by this way can activate NK T cells. So a bit more in detail, as most of you know, mRNA, whenever encoded in LNPs, enters in vivo in antigen-presenting cells like a dendritic cell. Whenever the mRNA enters the cytosol, it's translated into the corresponding protein and further degraded into epitopes, which are presented on MHC1 and MHC2. And in this way, I will focus on the MHC1. And this MHC1 presented epitope or peptides can activate the corresponding CD8 T cells. And by making use of this modified uridine, so modified nucleosides mRNA, nucleoside modified mRNA, we increase actually the translation and therefore the immune sensing. The second component of our formulation is the adjuvant, the alpha-galactosylceramide. And by co-encapsulating it in the vaccine, it also enters the cytosol, and by this way it can complex with CD1D molecules and activate NKT cells. These NKT cells can further then stimulate again the dendritic cell through the CD40 ligand interaction, and therefore upregulate the MHC1 presentation from the antigen of interest, and by production of IL-12 and other chemokines and co-stimulatory molecules enhance actually this axis here. So the alpha-gal-ser is acting mostly on maturing the dendritic cell and therefore actually indirectly enhancing the CD8 T cell responses. In addition to this, the alpha-gal-ser or the NKT cells by its way have also additional direct killing activities through the production of interferon gamma, and can also influence the activation state and functional properties of multiple other relevant immune cell types, as you think of NK cells and MDSCs. So now that we have the theory, we wanted to test our formulation, so the encapsulated alpha-gal-ser in the mRNA-LNPs in vivo, and therefore we first had a look at whether we can induce these NKT cells, and we performed actually an in vivo experiment in like 6 mice and injected several doses of our Galsomes intramuscularly, and also compared it to the same formulation of the highest dose but with no alpha-gal-ser, and looked at the amount or the number of NKT cells. And in the draining lymph node, we could see that there's a sufficient amount of NKT cells induced whenever our alpha-gal-ser is present, and this is dose depending. Further, we could look into their activation status to the PD-1 marker, and we could see that they are actively upregulated, and therefore this is also a dose relative effect. When we looked at the spleen, we couldn't see any difference in the number of NKT cells, but whenever looking at their PD-1 status, we see an increase again correlated to the dose, which was not present in the LNPs without the adjuvant. Looking at a shorter period of 24 hours, we could see that the alpha-gal-ser has its effect on the antigen-presenting cells by an enlarged expression of the mRNA. Here we used the Taiwan-1 mRNA, and we could see in several relevant immune subtypes that we have an increased expression of the mRNA when our alpha-gal-ser is included, which is each time in red, in comparison to the same formulation without the addition of the alpha-gal-ser. Looking at the maturation status of these cell types, we could see that our adjuvant actually induced an upregulation of the CD86 marker, which indicates the maturation of the antigen-presenting cell. Further moving on to immunogenicity studies, because this is the most important, the adaptive immunity for protection in vaccines against infectious diseases. We looked at an experiment where we compared again formulation without alpha-gal ser with the alpha-gal ser. Here we used mRNA encoding for ovalbumin, which is a model antigen, and we performed a prime boost vaccination experiment with 2 weeks interval, and after another 2 weeks, so at 4 weeks, we looked in the spleen at the antigen-specific CD8 T cells, so OVA-specific CD8 T cells in this case, and we could see a large induction of these cells for both the formulations. When looking at the later time points, we could still observe these responses. Although they were reduced, we were still able to detect a sufficient amount of these cells. Looking at their memory status, because we want a vaccine that is quite long in protection, so can induce strong protection. We looked at tissue resident memory T cells, and we saw a nice fraction at 2 weeks, but at 6 weeks we could even see that our alpha-gal ser induced here more tissue resident memory T cells in comparison to the same formulation without the alpha-gal ser. Now that we explored our formulation in using model antigens, we further wanted to test whether this formulation could be of advantage in context of vaccination against Mycobacterium tuberculosis, and therefore we formulated a vaccine encoding TB antigens. Here we chose for 2 well-known TB antigens, the AG85B and the EZ6. So we co-formulated, so we put two different types of mRNA in one LNP, and we formulated again a vaccine without alpha-gal ser and one with the alpha-gal ser. Performed a similar experiment to the short regimen from before, and here we could see that against the 2 targets, we were able to induce some nice responses in CD4 T cells. However, we were not able to detect any CD8 T cells, so only CD4. But later this became clear from literature that the combination of these antigens with this haplotype of a plaque 6 mice actually doesn't result in presentation of sufficient epitopes in context of MHC1 and therefore activate CD8 T cells. In addition to this, we could also see that, again, as a slide from before, that these vaccine-specific CD4 T cells were able to induce a certain type of memory. So we were able to see with our alpha-gal ser an increase in TREMs for both the targets. And this already at 2 weeks post boost, so at 4 weeks, while this was earlier not yet visible with the ovalbumin. So because we didn't observe any CD8 T cells, we wanted to further explore our formulation to the fullest, and therefore we used an alternative mice model. So we used a hybrid mice model, the CB6F1 mice model, which is actually a combination of the black 6 mice type and the bulb C. So it combines the haplotype B and D in mice. And by using this model and the same formulations, we were able to stimulate both CD4 T cells and CD8 T cells, in this case only for the AG85B, so not the case for EZ6. But this could give us a bit more, could let us show the potential of the Galsomes to the fullest. And here we also made a distinction between the Th1 response and the Th2 response in the CD4 T cells. And we could see that with our Galsome formulation, we induced actually a more Th2 balanced response, while the others, the conventional vaccines, which are they known for, is a Th1 response more. And in addition to this, so we looked here always at the spleen. We further also looked at the lungs in the same experiment. And interesting here is that with the CD4 T cells, we could actually see for a Galsome formulation that they were in higher numbers present when the alpha-gal steric was present in the formulation. And this is especially relevant for diseases that locate, for example, in the lungs, but this means that we have a more systemic protection, which is often required for these type of infectious diseases. So further, we did a proof of concept study using our Galsomes. And this we did in the Listeria monocytogenes model. Here, together with the VIB, with the Impens lab, they developed actually an immunopeptidomics pipeline. So they are able to detect novel antigens to an MS-based approach. And we then formulate these novel antigens into our vaccines in our Galsomes. And here we did, for example, a challenge study where we did a prime boost vaccination. We then, after 2 weeks, challenged them. And after 72 hours, we looked at the CFUs. So we counted the bacterial load. And with these novel antigens over here indicated, we could see really nice reductions over 95%. So these novel antigens were definitely working, but in this context, we wanted to know a bit more what is the effect of our adjuvant. So we did another study and we vaccinated here again with a formulation with one with alpha-gal sert and one without alpha-gal sert, and we here only vaccinated against the best of the novel antigens from the previous slides. We performed a similar study, and we could see that with our novel antigen without the alpha-gal sert, we already versus the control saw a nice reduction. But however, when the alpha-gal sert was included, we saw even a 45 reduction in the CFU. Interesting here is that we also took some extra controls along, which was the ovalbumin, so a irrelevant antigen. And this antigen didn't result in any reduction, but whenever there was alpha-gal sert included in the formulation, we could even see a drop here. So there was a quite a specific effect. So it means that this effect that we see here is likely a combination of the novel antigen and the alpha-gal sert. As Allegra also shortly already mentioned, this work is part of a European project which is called Tibaxerna. And in this project, we aim to perform this similar approach to this proof of concept. So first of all, we do antigen discovery with the VIB, so the Impens lab is included in there. They discover novel antigens for 3 important bacterial pathogens, which is the Mycobacterium tuberculosis, the Burley ulcer, and the Acinetobacter baumannii. And we then further include them here at Ghent University into the vaccines, and we hope that we can move the best vaccines of these hopefully to a phase 1 clinical trial in the next years. So with that being said, I would like to conclude. So from the work that we've been doing, we can say that our Galsomes broaden really the immune response of mRNA vaccines, and this is through triggering of NKT cells, enhancing the mRNA expression by itself, and also improving the DC maturation. And immunogenicity studies declare that we can induce more TRMs with inclusion of the adjuvant. And from the TB immunogenicity studies, we see some nice responses against 2 well-known antigens, the AG85B and the EZ6. However, we could not explore the signal to the fullest in Black 6 mice. We used hybrid mice models to look both at CD4 and CD8 responses, because we truly believe that the combination is necessary in context of TB. With our alpha-gal SER, we can further promote the formation of TRMs in context of TB, and we move more towards the Th2 balanced response. Further, we can also see that our alpha-gal SER promotes higher number of antigen-specific T cells, especially in the lungs, which is of interest here. And with our proof of concept study, we can say that our mRNA vaccines encoding novel Listeria antigens nicely protect in a challenge model, and this is also a synergistic effect of the novel antigen, so the mRNA-LNP and the alpha-gal SER itself. So with that, I would like to thank all my partners involved in this project from the Ghent research group of nanomedicines, as well as the people from VIB, from Impen's lab, as Karim Brekbot's lab at VUB for the mRNA production. So thank you.
Thank you very much, Ilke. Very interesting talk. So again, if you have any question, don't hesitate to write it in the Q&A. So I already have one from Karina Paskevich that asks, in the NTB vaccine, the Galsomes induce a more Th2 balance response. However, for this bacterium, a Th1 response is expected to be protective. Did you perform protection experiments and compare with an existing vaccine like BCG?
Yes, that's a really relevant question. So it's not that our Th1 response is completely gone, I have to say this. So it's still there. Next to that, we performed indeed a challenge study with these antigens, but as mentioned, these antigens are maybe also not ideal in the Black 6 model. And the partners that we worked with to perform these experiments, because we don't have a suited lab to perform these experiments, performed only the experiments in Black 6 mice. So I have to say we didn't actually see any protection at all there in comparison, to also BCG, which gave an expected response. So I think it will be important to further explore this using the novel antigens we will test from the Baccinologists approach. So it's still quite, it's actually too early to say now how our Galsomes will do in a challenge model, because we didn't use the optimal antigens for now.
Thank you, Ilke. So then we have Gita Malu that asks, have you used other lipids besides alpha-gal SER, for instance, mycolic acids?
We never tested such lipids. We had the idea to, but we didn't do it yet. We tested also some variants of the alpha-gal SER, and this one came out the best.
Thank you. So I will ask one more question, and then we can, Nicholas maybe can come back, and so I can ask one question each, since there are really a few for both of you. So which LNP works best with plasmid DNA? I think this one is for Nicholas as well. So do you think this strategy is good for extracellular bacterial pathogens? Clearly looks good for MTB and Listeria.
Yes, so indeed our main focus here is intracellular bacteria, because mRNA vaccines are known to induce a strong CD8 T cell responses if you compare them, for example, to protein-based vaccines. But it's known for extracellular pathogens that you want more an antibody-mediated response. But I have to say, I did also some research before on how we can design our mRNA sufficiently to get another type of immune response. For example, by adding trafficking signals in front of your mRNA and in the back of your mRNA. And you can, for example, also use secretion signals to boost more secretion of the protein, and in this way also stimulate more antibody responses. And in this case, it could be really of benefit for extracellular pathogens. However, there's still a lot to be researched in this field, as there's a lot of question marks still on how it will interact, how it will break down after translation into the cell.
Thank you, Ilke. So I will switch to a question for Nicholas. So Lars Frelin asked, do you think DNA-LNPs will replace the need of electroporation for priming a strong immune response?
So I think that the approaches can complement each other. I think that each have their role specifically. So in the context of some infectious disease vaccines, for example, where distribution is important and priming, specifically serological responses, may be important, that could kind of support a role for DNA-LNPs. Electroporation has been clinically demonstrated to induce very strong CD8 T cell responses. So I think both, like I was sort of alluding to earlier, as sort of like tools in a toolkit, so to speak, I think there's definitely context for both of those that would be important.
Thank you. So I will ask a question now to Ilke. So I would like to ask you, how do you assess the ratio? So you are co-encapsulating two different RNAs. How are you assessing the ratio that you have of the two different RNAs in the LNPs?
Really relevant question, because that's something also that we wondered. I have to say, in these experiments, I did it in a weight ratio. So I just added the same amount of microgram of mRNA whenever I formulate. But indeed, these two antigens have a different length. So there's different amount of molecules present in the LNP formulation. And as it's almost a double, actually, the length, it's quite a large difference. So we're testing now also how it's impacting whenever we add it in a molar ratio. But formulation-wise, it's the same. They result in similar quality of particles. But however, we don't know yet. It's on the plan to do that.
Thank you very much. So now I have a question for Nicholas. So it's a young Mao Yu. Have you by chance tested if the DNA-LNP integrated? This already, we had one similar. So then we have Rain Verbeke. Thank you for your talk. Interesting to see the comparison. Do you see differences in the acute toxicity between the DNA and the mRNA-LNP due to higher innate immune activation?
No, we haven't observed that. I mean, we're using in mice reasonably low doses, and we went up to 50 micrograms in rabbits and didn't see differences there. It's not to say that, a lot of the differences that we see in the innate immune kind of priming seems like almost specific different populations sort of increase or decrease. And so we think that perhaps is more kind of focused in generating that helping to prime the antigen-specific adaptive immune response overall, especially with those migratory DC populations.
Thank you very much. So I will continue with another question for Nicholas. Could you evaluate in vivo which cells are being transfected by the DNA-LNP?
Sure. So we did a study using luciferase and just whole body IVUS imaging in mice after an intramuscular injection. And so at least at the doses that we were looking at, primarily we can see the luminescent signal in the muscle. It's more local. Specific immune cells that are being transfected with the DNA, we haven't done those studies in depth yet, but that's definitely on top of mind, because conceivably, if we're upregulating different innate immune populations, absolutely there might be different populations that themselves are being transfected.
Thank you. And then Matti Salberg is asking, what is the key factor for the formulation of LNP DNA? And this I'm also very curious about. What is the secret to have DNA LNPs that actually work in vivo?
So I think that I'll answer that kind of similar to the way that I answered Dr. Liu's question, is that I think it has a lot to do with the NP ratio and better encapsulating DNA LNPs, because I think prior, using lower NP ratio formulations are not necessarily going to give the same biophysical stability for these lipid nanoparticles, and also on the DNA side, ensuring that you have, elements in the plasma that ensure high expression of the antigen. So you know that you're kind of optimizing both aspects of this, and then from a priming perspective, having the LNP and the inflammation that's associated with the LNP and the CGAS sting associated signaling associated with the DNA delivered by LNP, I think overall is going to help to prime the response altogether.
Thank you very much. Then I have a question for Ilke. So I would like to know a little bit more about how do you explain the unspecific effect of alpha-gal sera on bacterial growth for the diphtheria vaccine?
Well, it's the direct effects by production of interferon gamma. For example, as I explained in the beginning of my slides, it's not a specific activation. It doesn't need an antigen to activate. It acts all CD1D activation. So it's not like an epitope-specific stimulation.
Thank you very much. Then again, a question for Nicholas. Nicholas, you have still a few questions to answer to. So mRNA vaccine does not induce B-cell memory, a recent Nature paper. You showed that memory response in both mRNA and DNA vaccine modalities. Can you please explain? Thanks. By the way, good talk.
Thank you. Sure. So yeah, I'm familiar with that Nature paper as well. And for context, that is demonstrating that in humans, there's specific long-lived plasma cell populations in the bone marrow and as well as memory B cell populations that are not necessarily induced with mRNA-LNP immunization. And they compare that to some of the kind of common, more childhood vaccines. And so, yeah, that I think is a facet. Personally, I don't know that this has necessarily been specifically demonstrated in the literature, but is a facet of dose being given. So some of the mechanistic studies that have been done previously in the literature for nucleoside modified RNA, people are giving 30 micrograms, 10 micrograms, reasonably large doses, considering that in humans, we're giving in that, the 30 microgram to 50 plus range. So from a scaling perspective, I think that's one thing to keep in mind, because it would be sub 1 microgram would be the kind of dose equivalency. And so perhaps that is why we're seeing in mice is not necessarily translating to humans, is because like for example, in my study, I'm giving 2 micrograms of RNA. And so that already would be quite a bit. And so that's perhaps why myself, as well as others in the literature, can see those types of populations form robustly in mice, but not necessarily as you get into larger models and then through into humans.
Thank you very much. Then I have a TB question. Do you see a trans-amplifying mRNA vaccine for newborns a good strategy for replacing BCG and have a prolonged immunization?
Personally, I think we will never replace the BCG vaccine, as this is a vaccine that it's given to newborns on a large scale already the last many years. And it has some indirect effects against other pathogens as well in general, because of the well-known trained immunity that's present in the BCG vaccine. So they will never replace it, as it is also not an expensive vaccine. So it will be more a matter of boosting this BCG vaccine on a later age, I suppose. So in that way, it has and the self-amplifying part of your question, I guess, was whether this would be of interest.
Sorry, I'm
I
gonna
think it might have
reread
some.
it. Do you see a trans-amplifying mRNA vaccine for both, for newborn, a good strategy for replacing BCG and have prolongated immunization?
Okay, yeah, I think, yeah, first part is answered, but it's possible, yeah, but I think mRNA vaccines might be, yeah, also play a part in there. It will be a matter of boosting the BCG for sure.
Thank you, Ilke. So then I have another question for Nicolas. So which LNP work best with plasmid DNA vaccines? How many have you been trying? How many did you try?
So the majority of the work that I showed here in the slides today, I'm using SM-102, which is the ionizable lipid that's in the Moderna Spikevax formulation. We've actively been trying a number of different LNP formulations, and so I'm not going to comment on that answer just yet, but that is a active area that we're investigating.
Thank you. And another question, how do you compare the dose for DNA and mRNA, since one is double-stranded and the other one is single-stranded? How about the degradation of mRNA? Thanks.
So that is a great question, because it's something that we've thought about in the context of it's not necessarily going to be truly an apples to apples comparison, giving, let's say, 1 microgram of DNA LNPs versus 1 microgram of mRNA LNPs. And so we're not trying to match that in a certain way. We're just trying to say, okay, what is 1 microgram of mRNA as quantified versus 1 microgram of DNA? Because if we were to do these kinds of comparisons, we thought in either larger models or, hopefully if we can advance this even further, we're going to be comparing it the way that people have traditionally been, to doses of RNA that people have been using and such. So we haven't kind of changed the way that we're doing the study to try and better incorporate that, but we're just more to try and say, almost like we're doing this, what we would do in the clinic, and just kind of match it that way.
Yeah, thank you very much. So I will go with the last question, and then maybe you can share both your emails. So if anyone has other questions, they can directly message you, as I'm having a request of sharing, for example, the Nature paper you were referring to, etc. If you have like a slide with your email, please share it, so that's the one that were left behind can contact you directly, if you're okay with that.
Yeah, that's okay.
Thank you. So yeah, perfect. So it's in the chat, in case you can all check in the chat the emails, so that you can ask any further questions to them that were not asked today. So I have a last question for Nicolas. Would DC targeting improve activation of long-lived plasma cells and memory B cells, ensuring durable immunity?
Would dendritic cell targeting specifically? Possibly. I mean, I think that DC targeting could help to prime stronger CD4 T cell responses, and so that could kind of help in that context. One thing I didn't show for the sake of time is priming of GC responses, and so we can see nice germinal center responses being primed, and I think a lot of the associated kind of the durability associated with plasma cell responses or memory B cell responses likely will come from the germinal center. So having some additional signals there, or understanding how we can, continue to prime strong GC responses would be crucial.
Thank you very much. So thank you, Nicolas. Thank you, Ilke. That was a very nice webinar. Your researches are super interesting, and I wish you all the best for the future of your PhD. We had really a lot of questions, so that really shows how much your research is valuable. So thanks a lot. And thank you also to all the attendees, and for your many questions. Was very much appreciated. So we will meet for next webinar. You will soon find informations on our LinkedIn page, and also for those who are on the WhatsApp group, Thank you very much. Bye-bye.
Thank you.
Thank you, guys.
Bye.